columnar epithelial tissue, cuboidal epithelial tissue, squamous epithelial tissue. That's it. This is heart. What tissue? It would be cardiac muscle. If you write down muscularis tissue or muscular tissue, that's not correct. I want specifically on the microscope also what type of tissue is cardiac muscle. In this model, these are intercalated disc. These are striation, of course, nucleus, and then of course cardiac is only place in the body that has that cardiac muscle. Smooth muscle, all of the tubing inside of your body, all of the tubes, digestive, circulatory, the blood vessels, your trachea is surrounded by smooth muscle. One nucleus per cell, no striation. You don't see any striation. Smooth muscle. Cardiac muscle, what tissue is cardiac muscle? You see striation, nuclei are in the periphery. If you look at the cross section, you see the nuclei. This is cross section, nuclei on the periphery. You have slide of longitudinal cut. So you see the nuclei on the periphery. Do not worry about this, the neuromuscular junction and all of that. We have better slides to go over. Okay, so that's skeletal muscle, striation. This is a neuron. What is the name of the cell? Lab practical question. What is the name of the cell? Neuron. What is the name of structure? Axon. What is the name of structure? Cell body. What is the name of structure? Dendrites. And then what is the name of structure? This is axon terminal. Axon terminal. What is the name of the junction? Neuromuscular junction. Okay. And then you also on this model, the other model is a little bit better. This is node of Ranvier right here. Node of Ranvier right here. And of course you have myelin sheath, yellow, and this is the actual axon. So for this class, you should know these things. Axon, myelin sheath, nucleus, node of Ranvier. On the model right here, this is much better. You can see, I'll put a piece of tape right here. What is the name of the structure? Myelin sheath. What is the name of structure? Axon. Okay, what is the name of the cell? Neuron. What tissue? Nervous tissue. What is the name of structure right here? Node of Ranvier. You guys see the orange tape? You will see orange tape during exam. Okay, then cell body, nucleus, dendrites, axon terminal from another neuron. I hope I'm making some sense. So this would be presynaptic neuron. This would be postsynaptic neuron. Okay, nucleus of the myelin sheath. This is a myelin node of Ranvier. Okay, on the brain, I'm trying to go quickly so you guys. So the lobes, the cerebrum, the whole thing is called cerebrum with C. Frontal lobe, parietal lobe, occipital lobe, temporal lobe. So temporal lobe on this side, temporal lobe on that side. Then I'm asking you to know the cerebellum. Somebody will put it back together for me. Cerebellum, pons right here, and medulla oblongata. The white is medulla oblongata. They're supposed to be close to each other. And then the pons here. That's all. Olfactory bulb. Next year I added the list. But anyhow, that's part of the brain. That's all you have to know in the brain. In the eye, if you would, I think this is better right here. I'm missing part of the eye. So right here, sclera, cornea, most successful transplant is cornea. Iris, the opening of the iris is called pupil, right? Then I did not mention any muscle. Some of you answered the muscles of the eye. I did not mention anything. Underneath is a lens. Its lens is here, but it's missing. It's in the tray. Vitreous humor. The orange color right here is iris. The orange color is retina. And then right here, all of the vascularized areas is choroid. Look up the spelling, please. Okay, on this model, this is from a different model. Vitreous humor, and this is nice, beautiful retina, which they showed you rods and cones. Of course, over the years, it got dropped off. Bone. So, right here, compact bone, perforating canal, central canal, and that's all you can see on this model. The rest of it, I might ask you what is the name of that thing, is osteon. Haversian system, Volksman canal. Same name. Haversian and Volksman were the scientists who discovered this. Osteon. What is this model indicative of? Osteon. The whole model is osteon, not bone. The whole model is osteon. Of course, we know it's bone. Central canal, no perforating canal in here. These are osteocytes, canaliculi. This is lamella. One ring is called lamella. And then in here, again, the blue is osteocyte. This is matrix. All of this is matrix. Can you guys see this? And the orange is lacuna, the space outside of osteocyte. And then you have the canaliculi here, the central canal here, and the whole model is model of osteon. Heart. On the heart, you have to know 4 chambers, 4 blood vessels, and 4 valves. That's all I want you to know on the heart. 4 chambers, 4 blood vessels, 4 valves. Let's go. So right atrium, right ventricle, left atrium, left ventricle. That's 4 chambers. Okay, this one is missing. The part is missing. Okay, so that's 4 chambers. 4 blood vessels. You have inferior vena cava and superior vena cava. I consider as one. Inferior vena cava, superior vena cava. Okay. Then you have aorta, pulmonary artery away, and these are pulmonary vein. Pulmonary vein, pulmonary artery, aorta, superior vena cava, inferior vena cava, right atrium, right ventricle, left atrium, left ventricle. The 4 valves. The 4 valves is, this one is called right atrioventricular valve, left atrioventricular valve. This valve right here is called pulmonary semilunar valve, and this one right here in the left ventricle is called aortic semilunar valve, right here. Guys, if you during lab you don't know it, come to me and we'll look at it. That's it. That's all of the valves you should know. Skin. Again, I'm not going according to your lecture notes. Sorry about that. The skin, the stuff that you should know in skin is epidermis. What layer? Epidermis. What tissue? Cornified, stratified, squamous epithelial tissue. What layer? Dermis. And what tissue? Dense irregular connective tissue. What molecule? Collagen. What molecule? Keratin. What layer? Hypodermis or subcutaneous. Hair follicle, erector pili, the glands. This one is sebaceous glands, Sweat glands, and nothing else. What is the name of tissue here? Adipocytes or fat cells. That's it. I mentioned it. Okay. Model of intestine right here. So what layer? That would be mucosa layer, submucosa layer, muscularis layer. Muscularis layer. Submucosa, mucosa layer. Okay. Then what is the name of structure? That would be villus, villus, villus. You have 3, 4 of them in here. Inside of villus, what is the name of structure? That's lacteal. Lymph node. What tissue? These are a simple columnar epithelial tissue. The blue, what is the name of the cell? The blue one here, that would be goblet cell. And then, okay, lymph node. Here on the slides, you can see them much, much better. These are called duodenal glands. Right here is a cross section of it. Just look like your slides, but in here it does not look like a slide. Muscularis layer, smooth muscle. Circular muscle, longitudinal muscle. What layer of muscularis layer? Circular, longitudinal. And they both are smooth muscle. You do not have control over it. In this model, so you do have Oh, where are you? Here is epiglottis, the blue, right here. So it flaps over the trachea. This is esophagus. This is showing the digestive system, not showing the respiratory system, but the beginning of respiratory system is here. So this is epiglottis, esophagus, stomach. The folds of the stomach is called drugi. Also, these folds in the vagina is called drugi also. So these are the folds in the stomach called drugi. Do not worry about 3 parts of the stomach. Then this is duodenum, pancreas. Don't worry about spleen, liver, small intestine. Do not worry about duodenum, jejunum, and this is large intestine. This portion is missing. Okay, and anus, so on. In the liver, you should know gallbladder, and then because we need these later on in our life for gallbladder, I need to put my glasses on a little bit. Okay, great. So gallbladder, liver. This is called the cystic duct, hepatic duct, and this is called common bile duct. Common bile duct, I hope you guys can focus it. The common bile duct, hepatic duct, the green, because we need these when we talk about some parasites later on. Common bile duct, and then of course this is cystic duct from gallbladder. People remove the gallbladder, they're fine. Okay, and that's all you have to know there. On kidney, I don't break anything. Kidney. So, oh, sorry, that's okay. Well, thank you. On the kidney, you have the cortex layer, cortex, medulla layer, minor calyx, minor calyx, minor calyx, minor calyx, major calyx. You only have 3 or 4 major calyx. Major calyx, 2 or 3 minor calyx leading to major calyx. Then all of the major calyx lead into renal pelvis. Okay, and then ureter. So cortex, medulla of the kidney, minor calyx, major calyx, renal pelvis, ureter. They took a chunk of this and enlarged it for you guys. So this is a cortex, medulla. In the cortex, you have Bowman's capsule. In the Bowman's capsule, the white is Bowman capsule. The red inside is glomerulus. Okay, then outside, after Bowman's capsule, you have proximal convoluted tubule. These fold like convoluted. Proximal convoluted tubule goes to loop of Henle. From loop of Henle goes to distal convoluted tubule. From distal convoluted tubule goes to collecting duct. From collecting duct goes to minor calyx. From minor calyx to major calyx. From major calyx to renal pelvis, and then ureter, and then out. Okay. So cortex layer, medulla layer. Here they took a where are you? Right there. Okay. They took a glomerulus and enlarged it for you. So this is Bowman's capsule, glomerulus, proximal convoluted tubule, distal convoluted tubule, afferent arteriole, efferent arteriole, afferent arteriole, efferent arteriole. Okay, so that's all you have to know in the kidney. Okay, another model of heart and blood. Let's go over these quickly. Here is the lens. Lens of the eye is found in here. So here is another model of heart. So enjoy it. And then another stomach with drugi inside. Enjoy it. And then what you have, you remember there are 2, if you studied your notes, this is the other cornea that broke. Okay, so right here, this is a red blood cell. This is lymphocyte and monocyte. These 2 are agranulocytes. Agranulocytes, these 2. These 3 are, I call them 3 musketeers. These 3 are granulocytes. And maybe today or next Thursday, I should put the slide of these for you guys. The slide, microscopic slide of these. So the granules here are black or purplish. do not go by nucleus. The black or purplish granules, then this is a basophil. The granules are red because they pick up acid. They become red. It's called acidophil. Phil, it means love, acid, acidophil, or eosinophil. Eosine is the type of stain they use to stain these slides. And then, of course, these, they do not pick up, the granules do not pick up acid or base, so they're called neutrophil. These guys are 2.5 times of red blood cells. This one is the same size as red blood cells, the nucleus lymphocytes. Nucleus fills up the entire cell. And then the monocyte, the biggest of all, is 3 times of red blood cells. And in the monocyte, the nucleus is C-shaped. So these 3 do not go by nucleus, go by granules. These 2, you should go by nucleus and size of the cell. Nucleus fills up the entire cell on lymphocytes, and then here the C-shaped nucleus is very clear in monocyte. I'm done. I hope I'm done. Is there anything else I should know?